is that we need to be preventative versus reactive to our food and feed safety outbreaks. So when we talk about 𝗪𝗲 and prevention, how can we predict mycotoxins, right? They're so variable. They don't happen in every single load they produce when we speak about corn on a kernel to kernel basis.
Good morning, everyone, and welcome to this episode of the Feed Science Podcast Show. And I have with me a very special guest, Emily. Emily, how are you doing?
I'm doing good. How are you doing?
I am doing great. Emily, thank you so much for meeting with me this morning. And the first question that I have to you is if you could tell us a little bit about your professional experience.
Yeah, definitely. I can do that. So I'm Iowa born and raised. So I went to Iowa State University for all three of my degrees. So both my master's and my bachelor's was in animal science, where I focused primarily on ruminant nutrition and swine nutrition, did a little bit of poultry nutrition in there. And then I transitioned into industrial and agricultural technology for my PhD. And within that, I was in the Department of Agriculture and Biosystems Engineering. So I really focused more on risk analysis of different toxins, so on and so forth. And now I'm working for USDA ARS as a postdoctoral researcher in New Orleans, Louisiana.
Eastman serves veterinarians and nutritionists in agrochemical and animal health industries by helping them select, evaluate, and implement innovative nutritional programs. Eastman works with your team to customize your gut health approach in feed and water. Eastman's approach addresses nutritional and bacterial challenges and finds new ingredient preservation and hygiene solutions. Explore ways to accelerate and innovate your programs. Contact the animal nutrition team at eastman.com.
That's really good, Emily. And you got a really nice professional experience because when you were working in your undergrads, you were working with mycotoxins, and then you were doing some research with swine and cattle, right? I don't know if you could share some of that experience with us.
Yeah, definitely. So when I was in my undergraduate degree, I had a lot of opportunities to focus on swine nutrition. I worked for a lot of large swine companies such as 𝗵𝗶𝘀 and Smithfield. I also then transitioned into my master's thinking I was going to continue with swine, and I moved into ruminant nutrition. So when I moved into working with dairy cattle, I got a project like most masters do. You don't really get a say in what your project is. And it was working with trying to figure out how we can mitigate aflatoxin from moving from our feed-based ingredients such as silage and corn into the milk products. So how could we keep our feed and food supply chain from aflatoxin B1, which is our toxic molecule, to aflatoxin M1, which is found in our milk products. So with that trial, we really focused on looking at different dietary absorbance of how we can mitigate that aflatoxin. And then I really transitioned into looking at mycotoxins more on a corn standpoint for my PhD.
Okay, and that's really interesting. And when you were doing that trial, were you getting contaminated grain, or you were just using the active compound in those experiments?
That's a good question. So we actually did get contaminated aflatoxin grain, and we received that from the University of Missouri, which was already prepackaged. And we actually had to mix that contaminated aflatoxin grain ourselves into the corn. So I was there as a master's student wearing a hazmat suit, mixing in contaminated aflatoxin feed into our dairy rations. So you can imagine how that goes at a dairy cattle barn. But these trials are very, very important because we need to understand what can we do from a preventative standpoint to try to mitigate these things into our food and feed supply chain. We really need to keep humans healthy, other animals healthy, calves healthy, etc.
Yeah, and when because I'm always interested in because you also work in in the feed mill during the PhD, right? And I'm always interested to know more do you think that some of those mycotoxins are produced in the storage at the feed mill, or the majority of those mycotoxins comes directly from the field?
That is also a good question. So you did bring up a good point that in my PhD, I transitioned into working with the feed mill. So Iowa State has a brand new feed mill, the Kent Feed Mill Corporation and Feed Science Complex. So I helped create the food safety plan for that facility. But we believe that a majority of toxins are actually going to be produced in the field setting. You hear a lot about climate change and what is happening with our climate patterns, especially in the northern latitudes where our Corn Belt region is in the United States, especially Iowa, Illinois, Nebraska. It is getting warmer. When it gets warmer, we're going to produce more of our toxins, such as aflatoxin, 𝗟𝗶𝘀𝗃𝗃, or 𝗺𝗫𝗃𝗃, and we currently see a lot of fumonisin or 𝗻𝗃. But with this, it's not to say that these toxins cannot be produced in storage settings. We see a lot of, when we have this bad weather, high humidity, we'll have a lot of toxins being produced in grain elevators, in the bins, when the corn is just sitting there and we get some mold that might produce toxins. But it's not to say we have to be careful, because not all mold produces mycotoxins. We have to be testing for these things. If you look on the dairy cattle side, we really need to be watching our silage and our total mix rations. Silage we have to have good management practices because we can produce a lot of toxins in those scenarios.
That's really good. And, I see feed mills, they produce different species, and or they are going to be, some are commercial, some integrated. Does all the feed mills need to worry about all the type of mycotoxins, or they should focus on the mycotoxins that are of more importance for the species that they are producing?
That is a good question, because certain species are going to be more sensitive to certain type of mycotoxins. We worry about aflatoxin, right? Because that's a known liver carcinogen by the International Agency of Cancer Research. And that is the only mycotoxin that is actually mandated as an action level by the Food and Drug Administration. So for our species, when we're looking at aflatoxin dairy cattle, because of that transfer of aflatoxin B1 to M1. But if we're looking at swine, we're going to be more looking at deoxynivalenol or vomitoxin. It causes a lot of vomiting in pigs. And then across all species, zearalenone. It causes a lot of reproductive effects, such as abortions, infertility issues, embryo loss. So there are certain mycotoxins that we need to be on the lookout for, but it really depends on what region you're in, what does that crop year look like, what is the weather, and predicting that risk ahead of time for those species.
So something that is really important that you mentioned right now is prediction, right? Because, when I see mycotoxins, and when I go to feed mills, I always advise them to look, the reports of, mycotoxins from the area that they are getting their ingredients. But I think it will be better if we can predict, right? How the mycotoxins are going to behave, in the next harvest. And it seems like you did some of that research during the PhD. I don't know if you can share with us your findings there, or what tools are available.
Exactly. So that is a very good point, that we need to be preventative versus reactive, right? That is the whole underlying goal of the Food Safety Modernization Act, also known as FSMA, which was enacted in 2011, which kind of goes with FDA, is that we need to be preventative versus reactive to our food and feed safety outbreaks. So when we talk about FSMA and prevention, how can we predict mycotoxins, right? They're so variable. They don't happen in every single load they produce, when we speak about corn, on a kernel to kernel basis. And when we test, that's a whole other thing we can get into, testing and sampling for mycotoxins. So what can we do to be preventative? So to be preventative, we really want some sort of predictive tool that can tell us the risk in our given location and based on the species that we're feeding. So if we're thinking of a grain elevator or a feed processor or food processor, and we're creating a food safety plan that includes all of your ingredients and what that risk is in that given year, we want to try to understand how we can try to predict those toxins and what is happening in the field. So what we did in my PhD work is I collaborated with USDA ARS, and we created a predictive mycotoxin model for aflatoxin and fumonisin in Iowa corn. So the way that these models work is, it's artificial intelligence, big data, and we were able to create these machine learning methods to actually predict our risk of aflatoxin or fumonisin based on that current harvest year and what our weather patterns are looking like and what that risk would be to that elevator. So that way they're able to get, am I a high, medium, or low risk? And then they can put in controls into that place to try to mitigate those toxins to getting to that livestock or human food supply chain.
That's really interesting. And I was going to ask you, those weather patterns that you were talking about. When you are considering, those factors, the weather patterns, if it's hot and moist, do you also consider the maturity of the crop, or it's mainly, environmental factors?
Yeah, so right now we do want to look at the maturity of crop, that it would be a good input factor into the model. But right now, since it is so new, we kind of had the baseline that we needed to do. We're mostly looking at those weather patterns, such as, what is there a drought going on? What is our precipitation? What is the temperature? And then those can be inputs into predicting the risk. But we also added in a new factor, which we actually found in our Illinois predictive model, which was done by my collaborators. And we're finding that soil actually has some interesting effects of understanding how toxins are produced, especially in aflatoxins. So we're diving into this hole of looking at how soil properties might affect our toxin production.
That's really interesting, Emily. And, when you were talking about mycotoxins, right, they are very difficult to sample because, as you were mentioning, they will go from kernel to kernel. What tools or what kind of advice do you have, when regarding sampling?
Yeah, so this is definitely the culprit of mycotoxins is how do we obtain a representative sample? This is the biggest issue when it comes to mycotoxins, because a lot of times we really don't understand what's going on is because we cannot obtain a representative sample. And we know in Iowa, especially, this is the #1 corn producing state in Iowa. During harvest, these trucks are coming in and out of an elevator at a very rapid pace. Sometimes they're on the point of first receipt dumping the corn, and they're only there for maybe 3 to 4 minutes. And, lateral flow assays for testing for mycotoxins and sampling is going to take an average of 10 to 15 minutes, depending on who is doing it. So there are some protocols on how to sample. The main thing is you want to take multiple samples throughout the truck load. So you're going to pull and probe at multiple times when that corn is getting dumped into the pit. And we're going to grind and mix all of those samples so we're able to obtain a representative sample of what is going on. We really don't want to spot test those samples, because that's just going to be one time frame in that truck. And that doesn't really overshow everything that is happening in that picture. That's a good question.
That is true. And even like you're saying, when you receive a truck, it's going to take maybe 3 minutes, but the analysis is going to take 10 to 15 minutes. But at least you have the sample, and then you maybe can create like composites and then know what is the quality of the corn that you're getting. And I don't know, like if you could even segregate it by like the different suppliers, maybe, right?
Yeah, you really could, because that's another step. And you really want to have a mycotoxin control plan when we're talking about feed mills or grain elevators or cooperatives. You want to have a mycotoxin feed or mycotoxin management plan in place. So with that, in that step, you want to do ingredient verification from your suppliers and really understand what is coming from those suppliers and test those ingredients before you use them. Because once you bring them into your mill or into your elevator and they're already contaminated, you have to use those products or either store those products or determine what to do with them. So ingredient verification is extremely important.
And the problem is like typically those ingredients are more difficult to handle at the feed mill, right? Because then it's more difficult to segregate those ingredients once that they are inside the facility.
Exactly. You bring up, that's a good point, because when we're talking about corn, and that's where my experience is with mycotoxins in corn, you have multiple farmers coming from multiple different fields. So the traceability aspect of mycotoxins is just not quite there yet. I think we will get to a point where we can find traceability. A lot of my other collaborators worked with doing traceability with non-GM and GM products. But we're trying to kind of figure out how we can do that with toxins and how can we trace it back, because once they go to the elevator, they're all going to get commingled together into the bin, and then we kind of lose that contaminated sample. So if you do get a contaminated product into the bin or into the facility, you want to segregate it into another bin so you know where that product is.
Yeah, that's really good. And also the other challenge that I would like to get your opinion on it is, FSMA. Now, all the feed mills need to comply with FSMA, and then they have to analyze incoming ingredients. What will be your advice for feed mills to be in compliance? And I'm asking you this because I have sit in some meetings with the American Feed Industry Association, and then some of the issues that the feed mills have sometimes is that they don't sample enough the ingredients getting in the facility.
Exactly. So this is a tricky question because we all need to be in compliance with FSMA, right? Unless you're under certain amount, and unless you're a farmer. And farmers are exempt. So, a lot of these toxins are being produced in field and the farmer is exempt, and that's coming into the elevator. So that's really the point of first receipt, right? Like that's where we need to be testing and sampling. So in order to be in compliance, my advice would to be have a really strong food safety plan. So understand who your preventative controls qualified individual is, who your PCQI is, have a strong food safety team. Every single year, update that food safety plan. Don't let it go 3 to 5 years. Every single year, these crops are changing in fields. We need to be updating this food safety plan. And also have really strong CGMPs or current good manufacturing practices. And that should cover a lot of that FSMA compliance and regulation. Again, there is no specific mycotoxin management plan right now in the United States. There's nothing that each mill needs to do that is standardized. And I think that's really the goal of the future is what can we do to have a standardized mycotoxin management program at these grain elevators and feed mills.
Yeah, and even something that you mentioned is, PCQIs. And I think it's a good idea for feed mills to have more than one PCQI because sometimes, people changes jobs. And that way, they always have someone there that can help with, with the CGMPs and with the food safety plan.
Yeah, that is very important, is to have multiple PCQIs. It's just a training that you need to do. It is a very good training. It is long, but you can get PCQ certified for both animal food and human food. And I think that is really important from an industry standpoint, is to have these individuals trained so they understand what these toxins are, not just from a mycotoxin standpoint, but any chemical, physical, or biological hazard that is entering that facility.
Yeah, that is true. And the other question that I would like to ask is have you worked with any additives to mitigate mycotoxin risks?
Yes, I have worked with some feed additives, or also known as, on my side, adsorbents. I worked with the adsorbents with dairy cows, and we fed that with our aflatoxin treatments to see if it would mitigate, some of that aflatoxin moving from the feed to milk. It was actually really successful. We only added 28 grams into our diets. So if you think of 28 grams, that's a pretty small amount, for a whole dairy cow that weighs quite a bit of, weight. And from our trials, we were able to see about a 27% reduction of aflatoxin concentration from feed to milk. And that was able to get it below that threshold of what FDA says is, I think it's 0.5 parts per billion in our milk products. So I believe adsorbents do have a time and place, but I also think we need to really be more preventative versus reactive to these situations. So they're kind of a last-based resort of when you do have a contaminated feed product and you need to feed it out to your dairy cattle or your pigs, you can use some of these dietary absorbents. However, I do think we will have increase in toxin issues in the future. There's a lot of literature that has been published that it is going to just keep increasing. So I think we need to have everything in our toolbox to try to prevent these issues from getting to our food and feed.
That is true. And something that, like with feed additives, they probably are going to have like different specificity to different mycotoxins, right?
Exactly. So when we look at dietary adsorbents or specifically clay-based products like montmorillonite clay, silicoglycidyl clay, bentonite, those are really going to bind primarily to aflatoxin because that's the way that the molecule can fit and bind, and they can excrete it in either urine or feces. But when we talk about fumonisin and deoxynivalenol, it gets a little bit more difficult on trying to bind those types of mycotoxins. I'm not as familiar, but there is a lot of research going into different dietary absorbents to bind these type of mycotoxins. But it's not primarily clay-based products. It can be more like Saccharomyces cerevisiae, which is a yeast, or a lot of people are looking at algae-based products, more biological compounds on how to mitigate these other type of toxins. And then you bring up the issue of mass mycotoxins or multiple mycotoxins in one feed ingredient, and that's really where we're going to be going in the future, research-wise.
That's really good, Emily. And the other thing that I think it would be interesting to know is what you started at the USDA about 8 months ago, maybe. And so it would be interesting to know what are you doing right now, because, it seems like you have been staying in this line of research for a while. The other thing that I really like about your experience is that you started doing research as an undergrad, and I think we need, more undergrads getting involved with research. But I don't know if you could share some of what you're doing currently.
Yeah, definitely. So I actually started at USDA ARS as a postdoctoral researcher, and I just started less than a month ago. So I'm quite new into USDA ARS, but it's really exciting to see something different outside of academia because I was at Iowa State for 10 and a half years. So with the research that I'm doing at USDA ARS is I'm looking at controlling mycotoxins in alternative-based plant proteins. So that sounds like a mouthful, but I'm looking at how 𝗟𝗶𝘀𝗃𝗃, particularly that produces aflatoxin, grows in these alternative-based protein products such as chickpeas, lentils, green peas, also looking at corn and soybeans just kind of as a control. But in the future, I think that we're going to have both animal products and plant protein products to be sustainable. So we really want to keep both our animal-based products and our plant protein products safe from mycotoxins. So that's really the goal of this research, is understanding how these mycotoxins are produced in these proteins and how we can mitigate it from the supply chain.
And I think that is really important, Emily, because you see like some of some poultry companies, for example, that they are moving to all veggie diets, right? So, and we will probably in the future use more alternative ingredients. And when I talk about alternative ingredients, it will be like corn and alternatives to corn and soybean meal and like fats. So I think it's really good the job that you're doing, understanding mycotoxin risk in those other ingredients.
Yeah, I think it'll be unique from a standpoint that we're looking more, I worked primarily with animal feed and animal feed ingredients. That was my topics of interest. And now I'm really kind of focusing on human food. And, maybe some of these things could be used in animal animal feed byproducts in the future, depending on how popular they get. But I think we're seeing a lot of interest, especially from younger generations, with our alternative-based plant proteins and, other items such as like chickpeas and lentils and peas and of that sorts. So I think there needs to really be a lot of research at understanding what our toxin loads are. Do they even produce toxins, and what we can do to mitigate them? So this is just kind of this first step, this baseline at understanding do they get infected? And if they do, what can we do about it?
Well, I misunderstood at the beginning, but I think what you are doing right now with these ingredients in the human food side, we will be able to use a lot of that information in the animal industry as well.
Yeah, definitely. I think, I don't want to say it this way, but a lot of times the animal feed industry is a wastebasket, which is good because we are sustainable because we use a lot of byproducts that come from human food, such as bakery meal, all sorts of things that don't get utilized for humans can go to animals, which is if we're eating a lot more of these other plant-based proteins, a lot of these byproducts will go to our livestock. So we need to understand how we can mitigate the mycotoxins in this portion, especially for the livestock too.
Yeah, that's really good. And, one of, the last questions that I typically ask to our speakers is like, what kind of advice would you give to, either undergrads or grad students listening to the podcast? And it's mainly how they can use the tools in the university to prepare better for, to go out in the industry or academia and have a more successful career?
Yep, that's a good question. So I think my biggest advice to undergraduate researchers or undergrads coming into the university is never say no. Try to get involved. You can say, oh, I don't know about that. That doesn't seem like something I'm interested in. Maybe you'll learn that you're very interested in it, and just don't say no. Take on every single opportunity you can. For example, I went in, I was raised on a row crop farm, so soybeans and corn. My dad was an Iowa farmer, and we grew up with pigs. So I was a big pig girl. I was like, oh, pigs are my species. Mm-hmm. I got an opportunity in my master's to go into dairy cows, and I ended up loving dairy cows, and that ended up being my passion. From that, I knew nothing about feed mills and feed mill elevators, and I said yes to go to the PhD, focusing on feed manufacturing and how we can mitigate mycotoxins at the feed mill. And I ended up loving that too. And, now I'm here looking at alternative-based proteins, and I didn't think that would be something of my interest, and I'm finding that I really like it. So I think for students, just stay involved, get in clubs. Like if you're in animal science, like Block and Bridle is a great, club that you can get involved in, or different clubs, and try to, get those opportunities. Get involved in research if that's what you're interested in. Get involved in other activities. Yeah, just don't say no. Try to get involved.
That is really good, Emily. I really appreciate your time, Emily, and I have learned a lot today. Thank you so much for your time.